Hello viewers, so welcome to this lecture on the kingdom Animalia. The kingdom Animalia is divided into two sub-kingdoms, and the two sub-kingdoms are the sub-kingdom Parazoa and the sub-kingdom Eumetazoa. So we're going to look at the sub-kingdom Parazoa today, and in the subsequent video, we'll look at the sub-kingdom Eumetazoa. Organisms in the sub-kingdom Eumetazoa will be discussed later. The subkingdom Parazoa consists of animals that lack specialized tissues and therefore organs and a definite symmetry. The cells of these organisms are not organized into tissues that carry out specialized functions, and they also lack a definite symmetry. Parazoa includes animals of the phyla Porifera and Placozoa. The sponges in the phylum Porifera are the most well-known Parazoa. Okay, the subkingdom Parazoa has two phyla, the phylum Porifera and the phylum Placozoa. But the organisms of the phylum Porifera, that's the sponges, are the most well-known organisms of this subkingdom. Now I want to look at the subkingdom Porifera, which has the sponges. The phylum Porifera consists of unusual animals that lack definite tissues or organs. The organisms in this phylum are usually they are unusual animals, and they lack definite tissues and organs. The bodies of most of the animals in this phylum, that's sponges, are asymmetrical. They do not have a definite symmetry, and defining an individual is difficult. It's actually difficult to define any specific individual species in this phylum. They lack head or appendages, mouth or anus, and the internal organized structure characteristic of other animals. So the sponges lack head or appendages. They do not have a part of the body that can be referred to as the head, just as it is seen here. No part of these organisms at the left-hand side of the slide that you see now can be referred to as the head. Then they also lack appendages, just like appendages like hands and legs or even tail. They do not have appendages, and they also lack mouth or anus. No part of the organism can be referred to as the mouth of the organism or the anus. And internal organized structure characteristic of animals is also absent in these organisms. Their bodies are not really organized internally. At first sight, a sponge seems to be a little more than a mass of cells embedded in a gelatinous matrix. Okay, so they are quite unusual. These sponges are unusual sets of organisms. They number approximately 7,000 described species and inhabit all seas where they occur attached to surfaces from the intertidal zone to depths of 8,500 meters, that's 29,000 feet or more. They are usually found in all seas. In the oceans, they are marine, mostly marine organisms where they occur, where they are attached to the intertidal zone. That's the part of the ocean that's periodically covered and exposed when the tide flows and ebbs. Okay, they typically grow on rocks, dead coral, or anywhere else they can attach. They are usually attached to rocks, dead corals, or any other substratum. One family, the Spongelidae, is found in fresh water, and it has 150 species that live in fresh water. Adult sponges lack a definite nervous system and do not show conspicuous movements of body parts. They lack definite nervous system. Okay, they do not have a definite nervous system. There's no part of the organism you can easily refer to as a brain or the spinal cord or any other. They lack a definite nervous system. Just as we have said earlier, they lack specialized tissues and organs, and they do not show conspicuous movement. Just like plants that are stationary and completely sessile, these sponges cannot move. They don't show movement to them. They do not show conspicuous movement. Typical examples include barrel sponges, burrowing sponges, basket sponges, and bait sponges. And there are so many other examples that are enumerated here. So these organisms include the barrel sponges, the burrowing sponges, the, the burrowing sponges, the basket sponges, and the bait sponges. Now, this is a figure that's showing the sponges. And the sponges, just as you can see, they don't look like animals, but they are animals. Okay, they are animals. They're usually sessile, not moving. Sometimes they can be solitary or they can be in colonies. And their bodies have pores, their bodies are perforated. That's the origin of the name Porifera. The name of this sponge is the noble sea sponge. The scientific name is Pertodoris nobilis. Pertodoris nobilis. Now, we want to now look at the characteristics of organisms in the phylum Porifera. The characteristic of the sponges. The characteristics of the sponges. The organisms in the phylum Porifera have the following general characteristics. Their habitat. Now, I want to look at the habitat. All sponges are aquatic, mostly marine. Most sponges are usually found in aquatic habitats, especially, in fact, all sponges are found in aquatic habitats. There are no terrestrial species, and they are mostly marine organisms. There are just few freshwater species that are in the family Spongelidae. A typical example is Sponchilla, which is a species that's found in freshwater. Solitary, they are sometimes solitary, that's in isolation, or they can be in colonies, or they can be sessile. So these organisms are found in aquatic habitats. They are mostly marine organisms. Some are found in freshwater, and they can be solitary, they can be colonial, and sessile. Okay, they are attached to substratum. Sponges like warmer water. They usually like to live in warmer water. They are not usually found in cold water. Now, the second distinctive feature of the sponges is their body form. The body form of the sponges. The body form of the sponges. Their body is porous, is provided with pores. That's to say that their bodies have pores, openings. Okay, they have openings in their bodies, and they have these pores are very important in carrying out various physiological processes. The pores are of two types. They have two types of pores, and one of them is the inhalant pore, and the second is the exhalant pores. So they have inhalant pores and the exhalant pores. The inhalant pores are called ostia. The singular word for that ostia is ostium. Then the exhalant pores are known as oscula. The exhalant pores are known as oscula. So the inhalant pores take water and other particles into the sponge. Okay, and that's called the oscula. The ostia. The ostia takes in water and food particles and other materials into the sponge. Then the exhalant pores that are called the oscula are responsible for the discharge or the removal of water and waste products. Okay, now this is a figure that's showing the external and internal anatomy of a marine sponge. A figure that's showing the internal and external anatomy of the marine sponge. Now this is the osculum, okay, that takes water and waste out of the sponge. Then we also have the ostia that takes in water and food particles into the sponge. So water and food flows in through the ostia, and then they're expelled through the osculum. And the interior of the sponge is called the spongocoel. The sponges also have spicles, which are the internal skeleton that form the skeletal structure of the sponges. Then they also have choanocytes, which are cells that line up the interior of the uh, sponge, and they also have amoebocytes, which are also cells that are found within the body of the sponges. Now I want to look at the third characteristic of the sponges or the organisms in the phylum Porifera. That's their body symmetry, the symmetry. Most of the sponges are asymmetrical. Most sponges do not have a definite symmetry. Some are radially symmetrical. Some sponges are radially symmetrical. They can be cut into radial symmetrical parts. They want to now look at the fourth distinctive feature, which is the germ layers. The sponges are multicellular diploblastic animals. That is divided into two embryonic germ layers, this ectoderm and endoderm. Now their body layers are, they have two body layers. That's why they are said to be diploblastic. The sponges have two body layers, and that's why they are said to be diploblastic organisms. So their body layers are the ectoderm and the endoderm. The fifth distinctive feature is their level of organization. The sponges have cellular level of organization. They exhibit cellular level of organization. The sixth distinctive feature is their canal system. This system consists of pores and canals. Okay, their canal system. Three types of canal systems are found in sponges. We have the asconoid canal system, the seconoid canal system, and the leconoid canal system. The three canal systems in sponges are the asconoid canal system, the seconoid canal system, and the leconoid canal system. Now, the seventh characteristic of the sponges is the skeleton. All sponges possess an internal skeleton. All sponges possess an internal skeleton or endoskeleton. It may consist of calcareous or siliceous spicules or of spongin fibers of both located in the mesohyl layer. All right, so their internal skeleton can be made up of calcium carbonate, can be made up of silicon or of spongin fibers Siliceous spicules are said to be made up of calcium carbonate. Calcareous spicules are made up of calcium carbonate, while siliceous spicules are made up of silicon, or they can be made up of spongin fibers or both. Now, the digestive system of the sponges, it is intracellular and takes place inside food vacuoles as in protozoans. The digestion takes place within the cells and occurs in food vacuoles. They do not have a digestive system. The food is digested within food vacuoles as it is in protozoans. So they are very primitive organisms. Then the ninth distinctive feature is their circulation. Distribution of food from the ingested is distribution of food from the ingesting cell to others is brought about by wandering amoebocytes of mesohyl layer. Okay, so the amoebocytes are usually responsible for the distribution of food. They are responsible for the distribution of food. Respiration. Exchange of gases occurs by diffusion through the plasma membranes of the cells as in protozoans. So each individual cell carries out respiration. There is no respiratory system, and it occurs by diffusion through the plasma membranes of the cells. It occurs by diffusion through the plasma membranes of the cell. Now, excretion. Removal of excretory matter also occurs by diffusion through the plasma membranes of the cells, as in protozoans. Ammonia is the chief excretory waste. So the sponges do not also have a excretory system, just as it has been said earlier that they lack definite tissues and organs. And as such, the wastes are expelled from the sponges through the plasma membrane by diffusion. by diffusion, and they excrete ammonia as their excretory product. Their excretory product is ammonia. The 12th distinctive feature is their reproduction. Both asexual and sexual reproductions occur in sponges. Both asexual and sexual reproductions occur in sponges. Asexual reproduction occurs by budding and germ moulds. Asexual reproduction occurs by budding. Budding basically means when a sponge is formed as an outgrowth. Okay, so asexual reproduction occurs mainly by budding or by germ molds. In freshwater and a few marine sponges, germ molds or internal boards are formed. Okay, the germ molds are actually internal boards that are formed within the sponge. Sponges are hermaphrodite. Fertilization is internal. They are hermaphroditic. They have both male and female uh, gonads or reproductive organs in one species, and fertilization takes place internally. Now look at the development, which is the 13th distinctive feature. Zygote undergoes holoblastic cleavage. Okay, when the zygote is formed, it undergoes holoblastic cleavage, which is a complete division of the zygote. The zygote divides completely. The development includes a free-swimming larva and the amphiblastula or insecon or parasimola, which is found in Leucosolena for dispersal of species. So the development includes free-swimming larva and the amphiblastula or parenchyma for the dispersal of species. Now, I want to also look at the classification of sponges. Classification of sponges, organisms in the Phylum Porifera. The classification of the organisms in the Phylum Porifera. Sponges are classified into 3 classes depending mainly upon the types of skeletons found in them. Okay, the criterion for the classification of the sponges is based on the kind of skeleton they have. The classes are 1, the class Calcispongia or Calcaria. Then 2, the class Hyalospongia or Hexactylidae, and the class Demospongia. So these are the 3 classes of sponges. So I want to look at the class Calcispongia or Calcaria. The class Calcispongia or Calcaria. Members of this class bear speckles of calcium carbonate. They bear speckles of calcium carbonate. Their speckles or skeletons are made up of calcium carbonate. Species are either verse-shaped, compact structures, loose networks of thin tubules, or irregular, massive colonies. So they can be verse-shaped, they can be loose networks of thin tubules, or they can be irregular, massive colonies. They are mostly small in size and inhabit shallow waters of all seas, from intertidal regions to depths of 200 meters, that's 660 feet. A few species live in depths of 800 meters, that's 2,600 feet. So they actually found they are usually small in size, and they are found in shallow waters of all seas and in the intertidal regions to the depths of 200 meters, which is 660 feet. Then few species can be found in depths of 800 meters, which is 2,600 feet. They are filter feeders. Organisms in the class Calcispongia are filter feeders. They filter food from the water. There are about 400 described species in this group. There are about 400 described species in this group. Now here is a sponge in the class Calcispongia. It is called Clathrinia clathros. Okay, is an asconoid calcareous sponge. Okay, it's made up of calcium carbonate. The body of the sponge is made up of calcium. The skeleton is made up of calcium carbonate. The second class of the phylum Porifera I want to look at is the class Hylospongea. The class Hylospongea is also called Hexactinellidae. So we want to look at this class Hylospongea. They are also called the glass sponge or the Hexactinellid. So we want to look at these sponges. Skeletons of the sponges in the class Hylospongea are basically of hexactinal, that's 6 grade, and possess siliceous spicules and lack spongin. So unlike the calcispongea that have calcium carbonate as spicules, or whose spicules are made of calcium carbonate, the skeleton of those in the class Hylospongea is made up of silicon. They have siliceous spicules and they lack spongin. They lack spongin fibers. They are water-loving sponges. Their dried out skeleton often looks like dried out glass. Their dried out skeleton, when their skeleton is dried out, it looks like dried out glass. They are sedentary and range in size from 10 to 30 centimeters. Okay, they actually live in isolations and they range in size from 10 to 30 centimeters. Their cytoplasm doesn't divide into cells. Okay, their cytoplasm do not divide into cells. They are exclusively marine organisms in deeper waters or seas, depths from 25 to 850 meters. That's from 80 to 2,900 feet. Commonly fixed firmly to a hard surface, some species anchored in soft bottom sediments. There are about 500 species of sponges in this class. Okay, and examples include Hexactinella and many others. Now, these are the organisms in the class Calcispongia, the class Hylospongea. The class Hylospongea, okay, they are the class sponges, and just as you can see, they have that feature. Now, I want to look at the next class, which is Demospongia. The class Demospongia. The sponges of the class Demospongia possess skeletons of either 1 or 4-rayed siliceous spicules, possess spongin fibers, or both. So now look at the distinctions. The sponges in the class Calcispongia have skeletons or spicules that are made up of calcium carbonate. They have calcareous spicules. Those in the class Hylospongea are known to possess spicules that are made up of calcium carbonate. They're sort of silicon. They have siliceous spicules. Now, those in this class, now Demospongia, are also known to possess siliceous spicules. And in addition to the siliceous spicules, that's spicules that are made up of silicon, they also have spongin fibers, or both of them in one species. Skeleton is lacking in a few primitive genera. A few genera lack symmetry. A few species of these sponges lack skeleton. The sponges of the class Demospongia are the most abundant and widely distributed group of sponges. Okay, and they have about 4,200 species. This class has about 2,400 species of sponges. Now here is a figure we want to look at of the sponges. Okay, the figure of the sponges that of the class Demospongia. The class Demospongia. The class Demospongia is made up of these organisms. So I want to now look at very important features of this Demospongia. Okay, the organisms in the class Demospongia. Now, we appreciate, sorry for the H, I'm unable to display the figure. Now, thanks for watching this